Welcome to the fluorescent microscope. So before anything else, including using the microscope or even trying to open the software, we need to turn it on. So we're going to go under the table, and it's actually a 3-part system to turn it on. Make sure you get all of these parts or else it's not going to work the way you think it is. We're going to click this button up here. That's 1. Button down here. That's 2. And the switch down there is 3. With all of that, you'll see the microscope begins to turn on, and that's your cue to double click this software right here. So as it turns on, I'm just going to explain all of the parts that are attached to the system. It's very similar to a typical microscope with a few more things that make it very unique. The first thing is we have this thing here that pushes in and out, and you'll see that it has 2 stops if you hear those little clicks. When it's on its innermost setting, that's when you'll just look into these 2 eyepieces here and see what you're doing in here. You're not going to see anything on that computer. If we bring it out midway, then you can both see and hear with your eyes, and you can see it on the computer now. Now, when you're ready to take actual images of the samples that you have underneath, that's when you're going to pull it out all the way. When it's on its furthest setting, that's when you'll click capture and get whatever image you're trying to get. So if you've taken any biology course, you know these are your eyepieces, you know this is your stage, this is where you'd put your slide, and this is the light that would shine through to it. And then usually you might be used to seeing the knobs on the side for how you adjust it. Now the knobs are a little bit different on this system. We have 2 ways we can control it. The first way is on the screen here. If we click the X and Y axis thing here, you'll see that as I press these buttons, we can go up and we can go down. And you see that you can adjust it to be a fine adjustment or a coarse adjustment. The other way we set it is via these, and these are a lot more precise. So, you know, typically when you're using a microscope, if it's on a higher objective, you're usually going to have this slide be much higher up. So something I like to do when I put my slide in is I'll usually just bring it all the way up to be safe anyway. Then we have these nice guys here. We have a top knob and we have a bottom knob. And you're mostly only really going to have to navigate the top ones. You're not really going to have to worry about this one down here. So you'll see as I shift this here, the stage is moving up and down. This up and down. That's what this top knob is. You'll see this one as I move it. Let's show you from this angle actually. Is moving it front and back. So this knob is that way and this way. Back knob is up and down. Front knob is towards you and away from you. So I'm going to set it back to fine. I'm going to bring us back up to our top. And now we can look at the software. So the software looks a lot more intimidating than it actually is. This big square is where you're going to see all of the images as you put them under the microscope, and this is where you're going to navigate all of your settings. So you're going to see we have 2 windows up here. This is the one where you open your projects. It's also where you're going to create your new areas for any images you're taking. Make sure everything you make is named extremely specifically, including things like if it's a control, if it's an experimental group, things like what you stained it with. Is it ZNP1, which is a synaptic marker? Is it fluorescing at 488? Is it 594? You want to be as specific as you possibly can. Then we go to the acquisition one, and this is very important for actually taking our images. The images we take are in a Z stack, so you always want to have Z selected. What is a Z stack? It's when you have a thick or 3D sample, and instead of just taking a picture of one layer of it, you can get layers from the bottom of that sample all the way through it to the top. So it's giving you a very comprehensive view of the entire depth of the sample instead of just one layer of it. So you want to make sure you have that Z selected. Then we see over here, this is our exposure / setting. So you can see you can adjust your exposure using this bar or typing numbers in. Usually you don't want to go up so high that it gets to 1 because that means you're overexposing your sample. You want to keep it below that. So in these kind of 100 number ranges. Then we have these things that you can play around with to further adjust the image and make it better over here. Then we drop down here. This is how you create your Z stack. which I'll get into in a second when we actually have a sample on it. This window and your thunder window are going to be some of your most important. Finally, we have this section here. You'll see that if you click on user settings, we have a couple different settings that we can use, and each of them gives you different light values. So these light values come into play when you're doing your staining. Some of your stains are going to fluoresce green, some of them are going to fluoresce red, and that's going to determine what kind of lighting you want to use. You'll see that depending on the settings, let's say I click this one for example, we can have settings for both green and red fluorescing stains. If I do this one, we also get something called a bright field, which just shows you the tissue on its own under the microscope. We have one for DAPI. DAPI is one that shows under blue lighting. And then we have one called Morris Lab. Let's see what that one is. This is DAPI in the red. So lots of options. Typically for my projects, I tend to work with this one. But depending on your project, you might be using different settings. Now the last window we should know about is this process window. This one is very good for when you take an image and it's kind of cloudy and you want to make it a little bit more deconvoluted. That's when you can go to something called lightning and thunder process, and you can process it so it's a lot clearer. But that's a much more advanced step. Let's just show you how to actually image your samples. Now, I'm going to stage a sample here before I turn it on and show you what it should look like. Okay, so the first thing we're going to do is we remember that this top knob moves it either away from us or towards us. So we need to get that blue light focused in a way where it's on the samples that we care about. So I'm just going to move this so that it's where I want it to be. And now this is where this bottom knob matters, because this knob, as you'll see when I turn it, moves our stage side to side. So we're going to use that bottom knob and this top knob in order to get this exactly where we want it to be. So as you can see, these are specific to the stage holder and moving it front and back and left and right. So once I have it in the place I want it to be, I'm just going to look under the microscope and then I'll show you guys what I'm seeing. Okay, so this sample looks a little interesting. I was trying out something new where I micro dissected them before I stained them. So a lot of your samples are not going to look like this by any means, but if we look inside the fluorescence microscope, you're going to see we have two heads in there that are fluorescing green. Very cool. The only thing is though, you're going to see that on the screen, we can't really see that, and that's because this is on its innermost setting. We pull it out halfway though, suddenly they appear on the screen. So here is where we're going to start using this, the top button, making sure it's on fine, and we're going to be able to twist it left and right in order to bring these in and out of focus. So now that we understand what these focuses do, I've turned off the light just so we can appreciate what we're seeing on the screen a bit more. So now this is when all of these settings are going to come into play. You'll see as I adjust the exposure, these are either going to jump up in brightness or go down in brightness. So there's obviously an ideal setting that these should be at. The FIM, you'll see it's the same thing, where if I adjust it, we see slight changes. So what is the rule of thumb? Where do we want this to be? Usually I use this as a guide, and we want this to be within kind of like a reasonable range around here-ish. You don't want it to be super low, you don't want it to be super high. Around here-ish I usually say is pretty good. I also want to show you guys what it looks like under the different settings. This is it with the thing I stained for, which is ZNP1, but the bright field, like I said before, it just shows you the actual tissue. So if I click that, you'll see literally just the tissue on the slide. And then if I click the red one, because I didn't stain for this at all, it's not going to show you anything that we actually care about. The only thing we really care about here was what I stained for, which is the thing that fluoresces green. Now, the next step before we start imaging them is that we're going to want to set up our Z-stack. And what our Z-stack tells us is this is how early we want to start taking images within the thickness, and this is where we want it to end. So I clicked the trash to clear up any prior settings. And now what I'm going to do is I'm going to be using these knobs in order to set my Z-stack. All right, so right now they look all right. The first thing I'm going to do is take this knob and turn it counterclockwise this way until I stop seeing what I'm interested in. So let's say we're looking at this sample, for example, and this isn't the greatest option. So I'm just gonna show you guys using blurriness as a metric. I'm gonna keep turning it counterclockwise until the sample gets blurry. So let me do that again so you guys can see as I was twisting it. Let's get it into focus. So I would say this is maybe the earliest where I can discern things. This is an area I like to start. So I go over here and I click begin. Now I'm gonna start twisting it clockwise in order to get the other opposite, the other extreme bound in which I'd like to stop. So I'm gonna keep twisting it clockwise now until it goes out of focus again. So that's pretty blurry. Let me see if I can catch it right before. So I'm just toggling between clockwise and counterclockwise until I get the other furthermost end in which I can still clearly discern anything. And I would say this is pretty good. So I'm gonna go over here, I'm going to click end. Okay, so there are two settings that are very important to recognize once we set up our Z-stack, and that's the number of steps and the Z size. What these two values mean is that depending on the size, aka how thick or thin you want each slice to be, you're either gonna have more or less steps. Now this has its merits. The thinner the slice, the more information you're getting, but also the more steps that you're working with. Usually you want your step size, depending on your experiment, to be either 0.5 or 1. We've toggled between the two of them. So you'll see obviously if I set it to 1, my step amount is going to decrease significantly because they're a lot thicker versus if they're 0.5. Roughly, we like it to hover around like 70-ish steps. We don't want it to be too much. Just because I'm doing a demonstration, I'm not going to pay too much attention to this, but a lot of these settings are going to vary depending on the experiment you're doing. And the last thing you want to do is make sure that these settings are set. So we're imaging a zebrafish, so we want to select that. We're doing it in VectaShield, and all of this should kind of populate on its own. Okay, so now that we have our image, we have our Z-stack, there are 3 things we want to check before we begin imaging. The first thing you want to check is that all of these settings are set exactly as you want them. So your exposure amount is within range because these have been set properly. This isn't too high. We have our Z-stack set, and we have our step size and number of steps good. We've selected zebrafish, we have VectaShield, and all of this is set. Next thing you want to check is that this is in exactly the view that you want it to be in when it's imaged. And the third thing we're going to check is that this knob is pulled all the way out. Now you want to make sure the lights are off so that there's no outside light obstructing the view. And then, last but not least, you're going to click Start. Now note, you have single image, it gets one image, capture image, which means it captures it exactly as it is. But if you click Start, that's going to take your actual Z-stack. Now the final thing I want to show you guys is just that there's multiple magnifications, as with any microscope. The only thing is, since we don't have an actual head that we could twist on our own, we do it on the computer. Now you're going to see you can't change anything when we're live. Since we're live, we can't change it. Right now we're on 4x. We'd have to stop the feed and then click the next one that we want to set it to. Now usually you're mostly going to operate between 4x and 20x. Very seldom do you need 40x, and to do that you usually have to put water on the slide. Usually 20x is the highest you're going to go, and do not fret. You do not have to lower and raise the slide again when you're going up to a higher magnification. As long as this is in focus exactly as it's meant to be, when you click that 20x, it should fit right in without breaking the slide. And with that, that is how you use this microscope. Now, as with every other thing in this lab, make sure when you're done with it, you turn it off, you close the software, make sure you always save your data, but that's pretty much it.
Every video ends as every lab session should, with a good cleanup of your space. With that, thank you guys for watching, and good luck in the lab.